ancestral chromosome 4, we can see that the P-arm contains dog chromosome 13 and dog chromosome 29 sequence. And this region is actually highly conserved across carnivores. So here, this is cat, this is a ringtail, this is the raccoon, and this is the giant panda. And not only in carnivores, but it's also conserved in human chromosome 8. So this means that not only at the ancestral carnivore karyotype, but even at the mammalian ancestor, these regions are conserved. When examining each of the canid chromosomes that correspond to these regions, we're able to identify patterns of centromerization. So as I described before, the dog centromeres for each of these chromosomes contain repetitive sequence and are termed mature centromeres. When examining the homologous regions in red fox, which are here and here, they have been separated by a centromere. And so the question is this centromere a new centromere or is it an established centromere? And the way that we answer that question is by using these centromeric probes. And what hopefully is visible, we do not have red, or we do not have blue or yellow signal, which means that the centromeric repeats are not present at that centromere. So we indicate that this centromere is a newer structure. What is very interesting in this chromosome is that this region here, which corresponds with the centromeric region of dog, does retain these centromeric repeats. And that really suggests that that location may be in an activated centromere. So as the structures of the ancestral chromosome here underwent changes for the canids, we believe that this region, which corresponds to a mature centromere in dog, likely was reorganized. So the orientation may have changed. And this region has not had enough time to remove these centromeric repeats. So in effect, this is a newer structure. When examining the gray fox, these are also the same homologous regions. We can see that there is also not a centromeric repeat section at the centromere, which again suggests that they are neocentromere, that it is a neocentromere. What is interesting is that the region that is centromeric in dog, which is similar to here and here, still retains at least one of the probes that represent centromeric repeats. So I hypothesize that with this structure, perhaps this organization of having this previous centromeric repeat is possibly in the process of undergoing inactivation.